So today's speaker is Professor Rob Scott, standing right next to me here. He grew up in Hamilton, Montana, and received his PhD from the University of Texas at Austin back in 2004. His research is united by an interest in environmental influences on hominid evolution. Professor Scott has developed his understanding of hominid evolution through studies of fossil specimens in museum collections and his own fieldwork as part of international collaborations in Java, Turkey, Hungary, and China. He's done extensive work reconstructing the ancient environments of our early ancestors based on the analysis of the wear patterns on the teeth of fossil finds. This method has provided new insights into the diet of South African early hominins and suggested the importance of fallback food exploitation, and was published in the journal Nature. Dr. Scott has a strong focus on the natural environments in Western Eurasia about 5 million years ago, and is a leading expert in the analysis of fossil cattle and horses in the reconstruction of ancient environments. More recently, he has begun work on the remains of Homo erectus in Asia, including field study in Java. At Rutgers, he teaches 3 interesting courses, one just called Extinction, one on human osteology, to the bones again, and also quantitative mathematical methods in evolutionary anthropology. So, with that expertise, please give a warm welcome to Dr. Scott.
Picking up the mic?
Yes.
All right, excellent.
Okay. And we're going to try and lower the lights.
That would be fantastic, yes.
I just wanted the lights up for me so that I don't have to wear my glasses.
So first, thank you so much for the very warm welcome. But also a thank you from Rutgers. And thank you from the Center for Human Evolutionary Studies at Rutgers, because at the end of the day, New Jersey has one big major research university. We're a big state, and we're here to serve the people of New Jersey, to offer an opportunity for the undergraduate students, graduate students, but also really to be able to reach out to the community, and events like this we should be doing more of. So thank you very much for having me. Also, just a plug, the Center for Human Evolutionary Studies, which I'm associated with, we have speakers regularly. We also have a Twitter handle. It's at Ches_RU, CHES_RU. And it's a great resource now to stay up to date on what's happening in human evolution, both we live tweet our speakers, but also every time there's a new find, there's some discussion on the Twitter. So I don't know if any of you followed the recent fossil out of Israel at about 180,000 years ago that's maybe a modern human, but that's one that we profiled. So thank you very much for having me. Today, I want to talk kind of about 2 things. One, just a little bit about one of the times and places that I think is absolutely the most amazing and fascinating in human evolution. So that's about in the Pleistocene, about 1.8 million years ago in East Africa, where we have many sites, particularly in Kenya, Tanzania, Ethiopia, that preserve a really fantastic diversity of what we call hominin fossils, or our close relatives. And it is just, if I had a time machine, it's where I'd want to go, right? But I also want to tell kind of my own personal science narrative, because at the end of the day, science is done by actual people, and sometimes what gets studied and how we get from point A to point B is an interesting story in and of itself, and a little bit random. So, because of Darwin, this is my favorite page from his notebook. I think mainly because at the top, he just writes, "I think," which I would suggest is a good idea. And then he has these notes, which are a little hard to parse, but they refer to the branching diagram that he's scrawled in here. And he notes that the gap between A and B is immense, and so is basically making the observation that we can look at different species, right? And we can see some that are similar, more similar, and then we can see great gaps. And that this is a way not just of organizing them, but a way of organizing them that is consistent with this notion of them having common ancestry, of common descent, right? And hence the branching diagram. Now, my 15-year-old is really into memes on the internet. So the classic meme for human evolution is of the shambling ape-like creature sort of slowly becoming erect, and then, you know, we have a modern human. This is a vision of human evolution, and it's one that I would say that is just simply not consistent with the actual evidence that, in fact, with human evolution, we have a great profusion of branching, and at many times during human evolution, really all of human evolution except for very recent times, more than one species of closely related hominin alive at the same time, either in different places or in the same place. So I think Darwin's branching diagram is awesome. And this, from his notebook and the muddled musings, a great deal of thought. This is then what he writes. And he, you know, the affinities of all beings of the same class have sometimes been represented by a great tree. I believe this simile largely speaks the truth. The green and budding twigs may represent existing species, and those produced during former years may represent the long succession of extinct species. So we are a green and budding twig, and so are endangered gorillas, chimpanzees, orangutans. And then if we go back in time and we look at a branch of our family tree, and here we are in East Africa, about 1.8 million years ago. We likely have about 4 different, what most competent taxonomists, which is really how we recognize a species in the past, would say are 4 different species of hominins, more closely related to us than we are to chimpanzees, all alive at the same time, right? And to me, that just like is mind-blowing, right? These guys, and you can see from the skulls, we've got relatively complete material, they looked different. They would have had a different way of making a living on the landscapes of the time. And they likely would have encountered each other, right? And so to me, I don't know if you all have had this experience, you might go to the zoo, right, and look into, say, the gorilla enclosure, and they can come right up to the glass, right? And you can really look a gorilla right in the eyes, right? And what I think we see is something that is recognizable as kind of like us, right? It's not like observing an ant or a wasp or a lemur or a monkey in many cases. It's that gorilla might look you right back. And I always wonder, well, what's that gorilla think of me, right? And now our lineage diverged from that, leading to the gorillas millions of years ago, maybe 8, 9 million years ago. These guys are far more closely related to each other than, say, we are to a gorilla, and they would have been walking around that landscape on 2 legs, bipedally, doing their thing, but they would have encountered each other. And I always like to imagine, well, what would they have thought of each other? Not a question I can really answer. But I can, and this is what my research agenda is about, and my interest is about, is try to take what that landscape would have been like, what the environment would have been like, what the ecology would have been like, what kinds of foods would have been available, what the rest of the fauna that was there, what the predators were like, would have meant for the hominins. So I want to put the hominins in their environmental context. Now, a principal way of doing that, and a senior colleague of mine, Bernard Wood, who's at George Washington University, he once came up to me and he said, "Oh, Rob, you only study the second most important thing in human evolution, diet. And, you know, sex is the most important, right?" But diet is kind of a fulcrum, a place where environment and the anatomy and behavior of our relatives all kind of come together, right? If we cannot eat, we cannot get energy, we cannot make babies, we cannot survive, evolve, and so forth. So, for me, what I'm interested, I'm not going to show a slide that hypothesizes about how these different hominins were related to each other, right? They were, right? It's hard to know exactly how they were related to each other. We know they were closely related. For me, that's enough. And I think the far more interesting question is what did they do and how did they make their living? So here we have, this is the 37-33 skull. Sometimes it's attributed to Homo erectus. If we want to be more specific and take a particular bent, we can call it Homo ergaster, which is kind of the African Homo erectus. This one dates to roughly 1.6 million years ago. The whole species sort of ranges 1.9 to 1.4 million years ago, depending upon what you may include in it. And it has a cranial capacity around about 850 cc's, which is a significant increase, okay? So human cranial capacities can be 1050, 1100, 1400, they can be all over the place. They're more, but apes like chimps and gorillas, much less. And the other hominins that would have been alive at this, let's say 1.8 million year, would all have had smaller cranial capacities. So what we know is Homo ergaster has put energy into growing a reasonably large brain, okay? And it has made the trade-off that says it's worth spending that energy, right? Because it's going to get more energy based upon what it can do with the brain. And so then the question is, well, what do you do with the brain? And that means more complicated, flexible behavior, in my view. And so this is Sarah Lubick. She has just recently defended her PhD at Rutgers. She has worked for years and years at a site in Kenya, at Kubifora, that's about 1.6 million years ago. And this site it has long been speculated that based upon some red earth there and some sedimological observations that perhaps this was a place where there was controlled use of fire by hominids. And this would be pretty much amongst the very earliest evidence. It's not an open and shut and closed case, but Sarah's dissertation has provided now the best evidence that it is perhaps the case that Homo ergaster would have been beginning to, in a controlled way, use fire. So this is awesome, and Sarah is a great example of a Rutgers product moving the field forward. So what then does this mean about Homo ergaster? So this is not published yet, but based on a study I did at Rutgers with some colleagues over in food science at the time, and there's an awesome machine called the TIM. It's an artificial digester. So what we did is we took potatoes, some raw, some cooked, blended them up, and we feed them into the TIM, right? And then the potatoes are digested. So there's a mix of digestive enzymes in there, and this is a model of human digestion. It's one that we can really control, right? And so what we have here is a comparison of bioaccessible sugar, okay? Really the kind of energy that you could get out of food based on whether it is raw or cooked. And what you can see here is a rather, what I think, remarkable difference in terms of the potential for energy that you might get whether something is raw or cooked, okay? So key thing, right, if you're looking at science, you want to criticize, always look at the scale, right? So I can make things look really extreme. But this scale, it runs from 0, right? So it is truly the case that we're looking about, kind of double or more the benefit of cooking food, essentially. So this then, fire, big brain, cooking, more energy, makes a nice set of facts that are consistent with the notion that one of those hominins on the East African landscape, maybe not at 1.8, but shortly after, was out there controlling fire, cooking food, using tools. And you can see then that the brain is a principal adaptation for accessing food and for making a living on the landscape. So enough about Homo ergaster. So this is a very famous fossil. It's the Zinj fossil, or OH5. It was given the name Nutcracker Man. Okay, and this would be, this one's Olduvai Gorge, right? And would have been around at the same time. Okay, so Paranthropus boisei, again, that same range of time period. You can see it potentially encountering Homo ergaster. And what is great about this fossil is it provides a real contrast in morphology and form to other hominins, right? So if you look, you can see, well, the big observation is the teeth. The molar teeth are huge, right? And if you look up, these are the premolars. The premolars are also huge. The incisors are smaller, right? So you have what's called the molarization of the premolars, and you have a lot of surface area on these big, relatively flat teeth. So we call them the megadont hominins, right? The guys with the big teeth. And this is, a kind of observation that I love to see on a fossil because I can point it out to a 4th grader, I can point it out to an undergrad, everyone can see it, and it's like, wow, this is major, right? There is, and this is another species of the same genus, Paranthropus. This is Paranthropus robustus, which is found just in South Africa. So what we have is in East Africa with Homo ergaster, Paranthropus boisei, and then in South Africa another species of megadont homin, teeth not quite as big, maybe, Paranthropus robustus. So two sort of differentiated by region cousins. If you really want to get into nomenclature, I would call them the Paranthropids, which is great because it follows the rules for nomenclature. I think this is at the sub-tribe level, if I remember correctly. But it's also a great pun, right? Paranthropit, because the classic hypothesis about what they ate was hard objects or hard seeds, like pits, right? So there's this whole great suite of characters that we see in both Paranthropus boisei in East Africa and Paranthropus robustus in South Africa, that all tell a story about processing food and about eating and about diet, right? So one of them is this space here is the temporal fossa. It's filled in with red. From the sagittal crest, which is sticking up here, through the temporal fossa we have the temporalis muscle, which is one of sort of the 3 major principal muscles of mastication and chewing. And what we can see by the size of that temporal fossa is that those chewing muscles were big, right? So they would have been capable of doing quite a bit of work, okay? So we talk about Paranthropus as being adapted for maybe doing some kind of heavy chewing, right? If you compare this to a modern human, our temporal fossa is much, much smaller, right? And so we are putting way, way less into a big temporalis muscle and chewing. So I don't know, I always ask questions. So how how much time do you guys imagine humans spend chewing in a day?
A few minutes at most.
10 minutes. I mean, not very much, okay? A chimpanzee spends like 6 hours a day chewing, right? 49% of a waking day or something like that, okay? There's a whole series of reports, the human area research reports that goes to different groups and measures chewing. There's some flaws in the data because one group reported in there, when I like sort of parsed it, it was listed as 7 minutes a day of chewing, and I was like, no. Nobody chews only 7 minutes a day, and I was imagining like, are they on a liquid diet? What is it? Turns out, if you actually read it carefully, the description, it says, the women were up before dawn and had made a complete meal and fed everyone before observations began. So there was some missing time, okay? But the reality is we spend very little time chewing, maybe 4 or 7% of a waking day, something like that, okay? So far less, okay? And that fits with our sort of relatively small teeth, our cooked and processed food, and that's pretty significant. Compare that to a chimp, the gap is awesome. So something like this, right, would be spending probably more time chewing like a chimp, possibly even more, and doing it with these big, powerful muscles, okay? The face is orthognathic, so it's protracted a little bit. This provides more leverage for chewing. The postcanines, they're big, like I said. The enamel is thick. The jaw is broad in this transverse direction in a way that is different compared to many other species. The incisors are small, and then the zygomatics, which we have here, they flare out, and that's where the masseter, another major chewing muscle, originates. So we really have, just as with Homo ergaster, where the brain size is big, right? And that says something about adaptation. We have a very clear picture from lots of different elements of morphology that something is going on with the paranthropods. Now, it's one thing, right, to make the observations on the fossil, and from that come up with sort of hypotheses about, well, what were they using those for, right? And we have to remember that what they were using them for is not also always the same thing as why that morphology originally evolved, right? Because we have numerous cases in evolution where a characteristic evolves as a result of one set of processes and then is sort of co-opted for another function and purpose. So what we need to do, if we're really going to talk about the behavior of these guys and figure out what they were doing on the landscape, and if we figure out what they were eating, how often they were eating, it starts to have implications for all of their other behaviors, right? Other lines of evidence. So one of the classic, just absolutely classic lines of evidence is dental microwear. So this is microscopic wear on teeth. So as the teeth come into occlusion, right, and masticate food, they wear facets, okay? And eventually your teeth are completely worn out. You may have had experiences at the dentist's about that, right? But as that's happening, these wear facets develop. And on the facets, there are microscopic wear fabrics that develop. And so if you zoom in really close, on the scale of microns, you can see another kind of landscape on the tooth. Okay, so I spent about 3 years at the University of Arkansas looking at the microwear of primates, living primates, but also of fossils from South Africa and East Africa, including the South African Paranthropus. I spent so much time looking at those facets that I would walk home and I would look at the sidewalk and I would see microscopic wear on the sidewalk, right? And then I would dream about it, right? Sort of a kind of really intimate relationship that's, I don't know, it got tiresome after a while. Now, the classic early metrics were to see that wear, right? And say, oh, this is a pit, this is a scratch, and then to count those. And people had to do it. And it was hard to do, and it was hard to replicate. But the work was so classic that right around the time that I finished my PhD, before I started doing microware, my son was born, and I would write all day until about 2:30, and then I would come home, and my wife would go to work, and I would take over childcare duty, and that was nap time. So it was great. So I would lie down on the couch in front of the TV, and usually I'd watch Crossfire, right? And sometimes I'd get stuck on another channel, and Charlie's like sleeping in my arms. And so one day, for whatever reason, I don't know why it was, I got stuck on the public access channel, and it was a statistics course. And they were teaching the chi-squared test, which is a very basic stats course. And the example they used was the frequency of pits and scratches on Australopithecus and Paranthropus in South Africa, right? And they gave the numbers, and they worked the contingency table, and it was just fantastic, right? But that just gives you an idea of how, classic the work is. And so what was found, right, is that there was the appearance that the Paranthropus specimens had more pits, and the Australopithecus were a little earlier in time, but with a much more gracile and less robust skull and chewing architecture, smaller teeth, had more scratches. And if we then think about sort of roughly how perhaps microware forms, and these are SEMs, this is a mangabey at the top, and you can see from the scanning electron micrograph lots of pits, and the idea is you have those facets come down and crush, and as a result of that crushing, you get the generation of pits. Now probably what is causing the pits is not the food itself, but maybe some other grit or something that's in the mouth that is hard enough to cause the wear. But it is the food that drives the mechanics of crushing, and it's those mechanics that potentially lead to the pits. Similarly, here we have a gorilla, and what you can see on the gorilla micrograph is much more striated, scratchy surface. And so this is the kind of contrast that was classically observed between these guys. And the idea was, well, this is consistent with Paranthropus in South Africa being kind of a hard food feeder, maybe nuts or something like that, and probably then Paranthropus in East Africa, right? The same thing. So this is our classic interpretation, that we have kind of a dichotomous set of diets, right? Two specializations, and in particular, in Paranthropus, a real specialization for hard foods, and the portrait is of a creature that is specialized in diet, not really a generalist. Okay, so just briefly, because I just can't resist a little bit of engineering, what do we mean by a hard food, right? So what we like to say what the food was, but what we can really only say is what were the kind of the properties of the food. So this is a stress-strain curve. The slope of this part is the stiffness. Okay, so something that's very stiff is like glass. It takes a lot of stress to cause a fracture, so it's very steep. And then eventually you get to a point where there's, if the slope is lower, there's a lot more elastic deformation of the object. So the area under this curve is toughness, and it represents the sort of energy to ultimately fracture something. And then the slope here is the stiffness, the kind of stress that can be absorbed without deformation. So when we say hard foods, what we mean is probably foods that are both stiff and to some extent also tough. Okay, they're really mechanically challenging. Okay, so what does that mean in terms of actual foods? Well, here's the thing. Primates mostly will take high quality fruits if they can get them. We have specialists that have more folivorous diets, but then primates will also eat a huge and vast range of other foods. So here we have some Brazil nuts over here, insects, meat. This is what we would call an underground storage organ. Roots, rhizomes, tubers, okay? Both these guys and these guys, they're going to be hard. Some fruits are going to be hard, right? So something like that would have been the idea of what these guys are eating. So far, what we have is a a story of, we have megalodon hominin morphology is explained by specializing on food requiring high force that causes fracture generally with less deformation, something like that. And we can't say exactly what that food might be. But maybe emphasize that we have a specialist. Okay, so here is when I was fortunate enough to get recruited into the whole field of dental microwear. These are we moved from the scanning electron microscope to being able to actually create three-dimensional models of these wear surfaces, so see how deep things were and see everything in 3 dimensions. And we teamed up with surface metrologists at Worcester Polytechnic Institute and take these awesome high-res 3D renderings. These are photo simulations from them. So here's a capuchin with a very pitty surface. Here's a howler monkey with a very striated surface. Howlers are relatively speaking quite folivorous, lots of leaves in their diet. And what you can see is we have these differences in morphology. And from that, we took some metrics designed by folks at WPI, and then I actually ended up designing my own one to kind of measure the overall directionality of wear in a surface. So this has a lot of directionality because of those striae are all parallel. This doesn't, in a very systematic way. So we could quantify dental microwear more precisely. Okay, and what that meant is that in our ultimate study, right, we have less variation, less noise in the system to confuse things. And so we could make more specific descriptions of what the microwear was. So one of the things that emerged is perhaps the microscopic wear between, this is in South Africa, between Paranthropus robustus and Australopithecus africanus was less than we had previously thought. That perhaps human observers had been a little biased in picking out differences. And then when we sort of look at that quantitatively, we end up with a graph that looks like this. And so here we have the directionality of the wear texture, and then this is the complexity of the wear, just kind of how chewed up it is, how pitty, how complicated. It's actually related to the fractal dimension of the surface. So a higher fractal dimension is greater complexity. But the point is these are two sort of replicable measures that could be automatically generated from our 3D models that we got from the scanner. And these fit with differences in diet that we observed in, we could sort of separate living primates. And what you see here is that there's a huge area of overlap between the two species, right? But that some members of Paranthropus have very complex microwears. That there was sort of a two kinds of wear regimes that we might observe. And so what this led to was that we need to refine our hypothesis of differences in diets between these two species to account for this. Account for the overlap and account for the differences. So our first kind of scientific update, and this is published in 2005 in Nature, is that perhaps that megalodon hominin morphology that we see is not explained by specialization on a particular kind of food. But it may be that that particular kind of food that's driving the morphology was a fallback adaptation, was a fallback food. And we see this in all kinds of living species where during a particular season or period of scarcity, on some kind of time scale, animals fall back on a food that's not preferred. It's not necessarily the best thing to eat, but it's available, and it keeps them alive through that period of time. So our refinement was to suggest that the differences that we see, which are old and classic and exist, if we look at them with a better microscope in 3D, a more repeatable method, we can see more subtlety in those differences, and we can make this argument that perhaps it is, in South Africa, a fallback food that's driving the adaptations. And that, of course, then gives us a whole sort of different idea of what their behavior might have been. It paints this portrait of a hominin doing things differently at different times, of encountering the variations, seasonal or perhaps otherwise, in the environment. Now, at this point, right, so here I start the talk, I say, oh, East Africa, 1.8 million years, that's the place to be, and now I'm just going on and on about microware in South African hominins, right? So wait a second, let's get back to East Africa, right? So the reason that the South African stuff was studied is because that's just where my postdoc's supervisor sort of did work, had access, and that's where the first microware studies were done. They became classic, and the similarities in morphology between Paranthropus robustus in South Africa and Paranthropus boisei in East Africa made it like, oh, why bother with the East African stuff, right? So towards the end of my postdoc in Arkansas, I said to my supervisor, I said, well, what about the East African stuff? And he kind of like gave this exaggerated, well, I have all those molds. I guess I have to do them sometime. It will be so boring. We all know what the answer will be, right? So I went off to bigger and better things at Rutgers, right? And in 2008, Peter and colleagues did the East African stuff, right? They published them, and the paper comes out, and it was like, whoa! That boring study, this is what it looks like, okay? So what we see are lots of scratches, not lots of big pits, not hyper-complex, chewed up, ripped up surfaces, but sort of relatively finely scratched surfaces. The scratches do not align in any sort of particular directionality, so the measure of directionality comes out as low. But this is not at all like the South African microware. It just isn't, right? And it's, so what it says immediately is, well, whatever is causing the wear, presumably, you know, the diet, it's different in these two things that have similar morphology. So similar, so well described, so classic that that's really not at all what we expected. So what I would say is this is one of the most awesome things about science, is when we come and we hit this moment of like, well, that is not what we expected, and we're really, really sure we didn't make a mistake in our data collection. And so something else must be going on, right? So then we published work. Well, so here we have in dark, this is boisei. This is robustus in the gray. Here's africanus in the white. So boisei tends to be not complex with only a very few specimens kind of directional. So it's a very, very different microware pattern. Here we have, this is the whole framework. The size of the bubbles refers to how big the sample sizes are, right? And this is, and so each of these sort of represents a different sort of set of microware textures. Things like the lion and hyena over, where's my little cursor? Oh, right here. These guys consume more bone, right? The cheetah, lots of just flesh, less bone, right? We have our grazing bovids, our browsing bovids. So we have lots of good contrasts with living species. The chimp and the gorilla are sort of where we would expect them to be, and the two different subspecies of gorillas where we would expect them to be, pretty much based on what their diet is. And so what we have here is, here we have over, you know what, since my, here we have Paranthropus robustus over here, and here we have Paranthropus boisei way over here. So they're in a totally different world, right? Suggesting the kind of differences of diet, right? And then, after this, studies of the carbon isotopes incorporated into the tooth enamel of different fossil species are published. And these reflect reliance on foods where the photosynthetic pathway of the plant was different and fractionate the different isotopes of carbon differently. So C4 plants are typically warmer weather grasses. C3 plants are typically dicots and cool weather grasses, a different So different plant foods, right? And what we have here, well, there's the blue dots clear over to one side. That is Paranthropus boisei. All the others are other hominins, okay? And so what we have is a C4 plant signature in boisei. So the isotopic composition of the foods, right, in South Africa and in East Africa are different for these megadont hominins. So it is a real, completely alternative line of evidence that says, hey, what they ate was different. They were eating different foods. Even though the morphology is very, very similar, right, the foods are different. To help sort of nail this all down, when Peter and I and colleagues published the data on earlier Australopithecus afarensis, which is the species that the famous Lucy fossil is into, and we can talk about Lucy falling from a tree later on. And Australopithecus anamensis, which is a little earlier, had microwear signatures very similar to Paranthropus boisei. So we imagined a sort of a trend of the dietary similarity between these and increasing specialization in Paranthropus boisei. So the picture, the hypothesis that we sort of put forward is they may have consumed Paranthropus boisei in East Africa. In contrast with the South African stuff, was consuming maybe tough grasses or sedges, and maybe actually really specializing on that food. Okay, so this is a kind of Pleistocene cow hominin, right? So maybe papyrus. So instead of Nutcracker Man, perhaps Paranthropus boisei is the swamp thing. I love this picture because this swamp thing, he has this face that I kind of imagine reflects the flaring zygomatics of Paranthropus boisei. So that's an idea, right? So our update then is that perhaps megadont homin morphology is not a single adaptation. Okay, so this suggests that perhaps there was convergent evolution, so the same morphology being generated by specialization on different foods, which might say something about these guys maybe being less closely related, who knows? Or perhaps what happened is we get that suite of heavy chewing characters, those big teeth and all of that, because of one set of selection pressures. And then the species diversify and they're co-opted to do different things. So you might imagine a South African branch that is descended from a more specialized East African group on sedges or tough foods, co-opting that to be able to consume harder foods during some seasons, right? It's a possibility. The problem with that is, and what I would have liked to have seen is if the isotopic signature for the East African Australopithecines, I would have wanted it to be C4. I would have wanted it to be the same as Paranthropus boisei. And the answer is it's not. Okay, so boisei really sticks out. So it suggests that the East African form was, while the microwear signatures were different, the foods were still were similar, the foods were still different based upon the isotopic evidence. So what this suggests is that early on, 4 million years ago, perhaps a little earlier than that, we have the first steps towards megadontia, towards big teeth and all these chewing adaptations, probably involve one kind of food resource. We then potentially see later on specialization with Paranthropus boisei, and then just another branch in South Africa where we see a different adaptation. So when I was an undergrad and in grad school, at the end of every seminar we used to say, well, what do we need to know more? And we would always say, well, more fossils, right? More fossils is the classic thing. so much has changed. We can now get ancient DNA out of fossils. We have whole frameworks in more recent human evolution of how things might have been related, and complicated histories of like Neanderthals and humans interbreeding that has really changed our sense of things. Plus now we get lots of new fossils. They never illuminate things, they just confuse things, right? So, I don't know. The only thing I distrust more than the DNA studies that purport to solve it all, or when we purport to solve it all based on the latest, newest fossil, right? So we don't just need more fossils. What we really need to sort out what these guys were doing, what they were eating, is just new and better evidence. So this is sort of my conclusion is that paleoanthropology has been on a kind of a long, strange trip from a day when people could be naturalists and go out and make a few observations of interesting species in the wild, find some fossils, and make some hypotheses. And things have gotten more and more complicated. We now need newer and better methods so we can understand the evidence more precisely, and we end up doing weird things, right? So for example, the study I did with the TIM machine, artificial digestion, right? If wetland foods, C4 foods, were important for Paranthropus boisei, we need to look at analogs that might preserve similar kinds of evidence. So we have one that I thought was cool, was the manatee. So here are manatee teeth. They're big, they're flat. They have thick enamel and constant replacement of teeth. They do lots of chewing, right? And this other just totally blew my mind, right? So the manatee has a radically different cost. It's one of the, I wasn't even thinking about microware. I was looking this up in the context of, well, what are some examples of the costs of digestion to compare to humans? And there's not that many species where this has been done. Like the skink, the goat. The manatee happened to be one back in 1986, where there's data on the increase over resting metabolic rate during chewing, and it's just radical in the manatee. So the energy of chewing is huge. The energy of digestion relatively low, it would appear, right? So I can imagine some sort of mechanical similarities between what a manatee was doing and Paranthropus boisei was doing. So right now, there's an undergrad doing her senior thesis. She's looking at manatee microware. So what does that look like, right? And to give us another view on the subject. Bipedalism can be relatively energetically efficient, whether that's what drove it or was a consequence ultimately. A bipedal Paranthropus boisei may have spent less energy on locomotion, which might allow a little more energy to be spent on chewing, longer, more powerfully, use of more muscle mass for that. We also, and this is my final slide, and it will be my final pitch, we need to understand better the exact mechanisms of how microware forms. So there's a whole, didn't really get into that in this talk, but there's a whole debate on how and or to what extent food is really influencing microware. So currently, my big project, which is just starting on Tuesday, we feed our first experimental pig. There's going to be 12 groups. They're going to get all these different diets, including, I didn't put the acorns here. Including acorns. There will be acorn-fed pigs. These are sort of a hard food. And what we'll be able to see is, based on exactly what the diet was, right, what are the microware textures. And this will hopefully be very illuminating. It has created all the problems that really happen in science, or these bizarre, mundane, weird things. But it turns out that for the M1 to be an occlusion so we can get wear facets, the pigs have to be older than they normally are when they're butchered. This means they're bigger, which means the butcher, the standard butcher that the farm at Rutgers uses, cannot fit them in whatever device it is that they use to like remove the skin and begin butchering. So we have to find another butcher. This is apparently hard to do. Now the farm can process them themselves, and these pigs are also far enough along that they're fattier, so most of what will result is sausage. And this is actually like a super ethical way to do this. We had another experimental design where we were anesthetizing them and putting them through all sorts of horrible pain. This is just taking the standard set of pigs that are run through the farm and actually growing them a little longer, and then, you know, they get turned into meat. So the farm can create sausage, but then the sausage will have to be moved, will have to be sold. And I naively said to the farm managers, oh, I'll just get on Facebook and get my colleagues to buy sausage. And he's like, no. You are talking a lot of sausage. So I may be sending an email to our board at Red Bank Humanists. To the extent that you eat sausage, we may have to move a vast amount of sausage. So thank you guys very much, and if you have any questions.
All right, everyone, so we'll do our Q&A, and so thank you very much, Rob. And I'll try and see who handed up and get around to everybody. Give everybody a first shot before we do a second turn at the mic. I guess I'll go with convenience first and then work my way back.
Thank you very much. I have 3 questions for you.
Uh-oh.
The first relates to the picture of the, I think it's the Paranthropus, one of the 4 skulls you showed in the beginning.
Right.
And I noticed a very prominent midline ridge extending from the surface of the skull.
Yes.
And it looks as though it might have probably been a muscle attachment.
Yes.
Do you know whether the temporalis muscle extended up to that ridge?
Yes, so that's the origin of the temporalis muscle.
Right.
So that's what's great about this, right? Where is that? That's what's great about this is that you have this concordance of like 2 morphological observations. So the temporal fossa, right? Big temporalis.
Right.
Sagittal crest, big temporalis, right?
Right.
And you see a large sagittal crest in gorillas, for instance. You see it particularly in species that are large, and that's because sort of as a result of allometries, you don't have relatively as much skull surface for the temporalis to attach to. So you often see a big sagittal crest in a male, and in a smaller female you may not see as large a sagittal crest. But it's definite, yeah, it's temporalis.
The second question I have is that when you showed the 4 skulls in the beginning representative of different species who existed at the same time, is it known whether they could intermarry? Could they be
Interbreeding, yes. We don't know. We don't know. So we don't, in this case, have any positive way to know. So classically, the idea of the biological species concept, which is a bit flawed, is, well, put 2 things together and can they breed and produce viable offspring? A graduate student was just telling me about a narwhal and beluga that produced offspring. There's like a 100-year-old skull. They did the DNA on it, and it's a cross between those very different whales, right? So it's hard to know what was possible. What we do know is that in later evolution, hominin evolution, Neanderthals and what we call the Denisovans were able to interbreed, and they had pretty different morphology, but they were more closely related than these guys probably would have been. Homo habilis and Homo rudolfensis, some people say they're the same species, in which case you could make a really good, in which case they would have been interbreeding, and maybe even if they were different species, maybe they were interbreeding. There is also evidence from the genetics that suggests that something that diverged back at about 1.8 million years ago, that lineage much later on introgressed and interbred with later hominins, which would suggest the possibility this guy and this guy interbreeding, I would say no way. And I would make that argument because, look, the adaptations are so completely different. They're just doing completely different things. They're not going to encounter each other in that way at all, and the morphology is very different.
The third question I have is totally different. I'm just curious, I see all these people working in Africa in different countries where you know that the political situation is usually unstable, there's a lot of violence. How are these teams of anthropologists and whatnot treated by the local governments and by the locals when they go there to do their work?
You know, every country is different, right? And it changes in every country. I mean, I don't work in Africa myself. My colleagues always report, you know, really positive things. In general, the extent to which there's a structure of a museum in the country really helps. So the Ethiopian Museum, the Kenya National Museum are fantastic. You know, Tanzania, in terms of having a research museum infrastructure, is maybe a little less developed. People work there very successfully. Those are relatively good places to work. A lot of my work's been in Turkey. It's kind of hard for me to go work in Turkey now because of the politics. Erdogan is not friendly to academics, right? The place that I really love to work would be in Syria, near the Syrian-Turkish border. I mean, that would be really an awesome place to go. I know there are sites, paleontological sites, that might be interested in Turkey. There's probably sediments in Syria. If we were looking for apes that are sort of along a potential migration or range extension that's sort of relevant to Africa, that would be really cool. I don't think I'll ever... Well, I'll be really old. Let's hope that someday we can do work in Syria, right?
Yeah, I'm sure it's very hard to study saliva and the effect of saliva from fossils, and I don't know exactly, and that does, in fact, affect what I eat all the time, hard or soft, is the saliva that I produce. So is there differences between species, or what assumptions do you make?
This is a fantastic question. This is a great question. So saliva does lots of things. So one, it's a medium, right? That's sort of lubricating, and it's involved in the formation of the bolus of food. So when I talk about the challenges of figuring out how microair forms, a naive notion of it's just the food as we see the food interacting with the molars is wrong, right? What's really interacting with the molars ultimately as we're chewing is the bolus, and saliva is critically important in that formation. If we look at, say, the cooking experiment that I did, right? Actually, the original plan, what I really wanted to do there, was I wanted to alter the composition of enzymes in the feed of the machine. So when you consume any starch, we produce amylase in our saliva, which starts to break it down. And the amazing thing is that there is a huge range of gene copy number variation for the amylase, the salivary amylase gene. So some folks in this audience may have 1 or 2 copies. Others may have 8, 9, or 15 copies. Okay, and this tends to correlate with ancestry and whether it was a high starch-eating ancestry. And it also seems to correlate with how much amylase is produced. So there's a genetic relationship there. So some people are going to get a lot more digestion of potatoes, for instance, done just during chewing, right? And so saliva is important. We wanted to vary the enzymes in the TIM machine. It's a little tricky. And those runs take an entire day, which is sort of...
So do you make assumptions then? Do you say they're all the same, different species?
So yeah, this is science, right? This is something we have to do. This is why I need really good graduate students who are willing to take these questions on and think creatively about like, well, exactly how are we going to study that? So I have a student who just finished, Marika Yaniak, who does digestive enzymes, and she has really great stuff on insect eating and primates. And she has some saliva samples of different primates. So she is going to look at the question of what's the variation in amylase in different primates, as she goes forward. So, yeah, no, that's a great question.
Have you ever presented this material to a creationist audience, and how did it go?
So that's an interesting question. So, okay, so I did my postdoc in Arkansas, right? I was a graduate student in Texas, and here I am in New Jersey. So the undergraduate student body is very different. I mean, there's a regional difference. And so my postdoc supervisor said, I lectured on evolution, and half the kids think I'm stupid and half of them think I'm a liar, right? It wasn't quite like that in Texas, I think the nature of the school, so there's regional diversion. I don't really encounter with students, many issues, right? I have lots of interesting conversations with Uber drivers all over the world. And let me just say, from my perspective, I don't emphasize a conflict between evolution and people's faith at all, right? So I heard somebody asking, what's the best argument in favor of like atheism or something? And I think the best argument is it's my business and that's what I think, right? And inevitably people get to make their own, draw their own conclusions, right? About what their observations of the world and how they interact with that and make meaning, matters. And this is what I would tell students. It's what I tell creationists. I don't think there's necessarily a conflict at the end of the day between evolution and faith. I mean, you can move there is a conflict of fact between a 5,000-year-old Earth, right? That's not the case because we can date rocks, right? The universe appears to be about 13-something billion years old, so forth. These are the sorts of things. But I don't think, accepting those facts necessarily means to challenge people's faith, and that's usually what I say, when I have that conversation.
So our ancestor, probably Augustus, clobbered all those others and conquered Africa first, and then sprung out from there into the world and conquered the world after that. How about those fossils just found in Greece and Bulgaria that were like 1 million years old, 2 million, and then the 100,000 one in Israel, was that a Homo sapiens?
Okay, so let's see. So Greece and Bulgaria, the recent stuff that's come out, which is actually sort of old, if that I would think of would be there's some apes, which is older at 8 million years, right? Okay. The very recent find, the one that was in the New York Times a couple weeks ago or whatever, the Mislia, right? In Israel, I mean, it's not a full skull, but there's a good sampling of teeth. The teeth do appear to be like those of modern humans and not of Neanderthals, which tells us that something that was anatomically Homo sapiens was there probably around 180,000 years ago. That's interesting, right? The things to keep in mind is just because something was anatomically like us, it does not mean it was behaviorally like us, right? Nor does it mean that it represents the migration that is ancestral to us. It may not jive with the genetic evidence because it may be, a failed migration, right? So we may end up with evidence from genetic models that suggests different migration times, right? To me, it's not as entirely unexpected as we might have imagined. The apes in the Bulgarian ape, the date on that is a little problematic because it was a surface find. And some people suggest, I mean, everyone likes to suggest that when you find a megalodon ape in Europe, maybe it is ancestral to hominins and there was a migration back into Africa, which is possible and is fun to talk about, but we don't have. Until we go to Syria, right, we don't have good evidence.
Talking about fallback diets,
Yes.
are you aware of any studies done on the panda bears, for instance, which we do know that originally developed as a carnivorous species and then evolved into exclusively bamboo shoot
Yeah,
domesticator? And I have another question unrelated to the first, which is, can you outline briefly the relationship between what's currently known about the human species, not the hominids, but the modern humans, like Neanderthals, the sapiens, the Denisovans, and the Floresiensis from Southeast Asia?
Okay, yeah, sure. So pandas are cool, so one of the first things I wanted to do, actually, and I never really got the chance to do it, was do the microwear of bears. So I have colleagues who've done that, right? And panda microwear, you know, does differ from other bears. My sense is maybe the panda teeth are really complicated. There's a lot of facets. So when I was working on that, there was some, depending on what facet you look at, you get different signals. And bamboo is a very unique resource with unique properties. But those are the kinds of studies that are really useful in sort of working out what microscopic wear means in diet. I have a colleague at Vanderbilt, Larissa DeSantis, who has an absolutely fantastic study on koalas who eat eucalyptus, and they have some of the most beautiful, striated, scratchy microwear imaginable. And eucalyptus doesn't have phytoliths, which are the little silica bodies that might cause wear, so that's really interesting, but the food is tough, and we think the eucalyptus gets coated with dust, so they're masticating this tough thing, and the teeth have to kind of slide by each other, and there's dust caught in there. As far as talking about, so Neanderthals, Denisovans, Flores, in terms of diet, right, the ones where we can make, you know, judgments about diet would be the Neanderthals and modern humans. And in both cases, we're immediately, from my perspective, in the complicated situation, these things have very sophisticated cultures and they can eat lots of different things. And so if we talk about Neanderthal diet, I mean, yes, they ate meat, and yes, they ate vegetables, right? They ate everything, and probably different groups of Neanderthals ate different things. So that's, you know, what I would say about Neanderthal diet. There's lots of other interesting questions about how those guys are related to each other in the history of populations. And so just a brief, we have some kind of we have a split, right, between the lineage that leads to us and the lineage that leads to the Neanderthals and the Denisovans that probably occurs in Africa. It may be that very shortly after that split, there is a migration into Europe and Asia ultimately, or possibly not. There are fossils that possibly could be sort of the Neanderthal Denisovan population, or maybe they hung out in Africa and came a little bit later. And then we would have a split in outside of Africa between the Denisovan and the Neanderthal lineages, with the Neanderthal lineages ultimately moving east. But also, before that, we have Homo erectus in places like Georgia, China, Java, and so forth. And we have Flores, way off on that island. And Flores is weird, right? So those were earlier migrations. And then there's some genetic evidence that there may have been introgressions into, say, the Denisovan population from possibly a Homo erectus derived population. as far as Flores, it could be Homo erectus derived. Some people have suggested it is derived from an even earlier split, right? So
How do you explain that there is no Neanderthal DNA in any of the African population in the state?
because that population moved out of Africa, and it was only later on that the population leading to us moved out of Africa. So what would have happened is, in Africa, there would have been no opportunity to interbreed with Neanderthals, and that it's as an African population moves out of Africa, it encounters local populations of Neanderthals, and that's when we get interbreeding. And that kind of interbreeding actually makes a lot of sense, because it's always good to interbreed with the locals. Because the locals have immune systems that have become adapted to the local pathogen landscape. So we have lots of evidence for selection against Neanderthal genes, and then we do have evidence for selection for Neanderthal genes. And a lot of the selection for Neanderthal genes is in immune genes. So I have arthritis, right? It's autoimmune. I think it's probably the legacy of my Neanderthal DNA. You know, it was useful, had a useful immune function, and it's a misfit for our modern environment. Probably because I never had worms as a child. Right?
Any other questions from the end?
Could you just define the term introgression that you've used
Ah,
several
yes.
times?
Thank
I'm
you.
not sure if I'm understanding.
Thank you. So we have interbreeding, right? And so introgression would be the sort of more formal description of when some genes, as a result of some interbreeding from one population, introgress and move into the other population, right? So the history of Neanderthal modern human interbreeding resulted in an introgression of Neanderthal genes into our modern human population. And it went the other way too. So we have Neanderthal fossils there's ancient DNA from Neanderthals that apparently interbred with modern humans, that have some modern human DNA, and that Neanderthal population appears to be unlikely to have been later interbred with modern humans and is not related to other Neanderthals, right? So it's just a way of saying some genes, due to interbreeding from one group, made their way into another, at some frequency.
And I guess it also means that it's become characteristic of that other group. Those genes became
They moved in, right, due to interbreeding. Then there was a history of selection for and against some of them, and we can now recognize them now as a part of the genome of that group. And what we found is that introgression is more common than we thought. It means there is hybridization between things that we would consider different species.
Could you give us 20 seconds of sanity on the idea of modern humans embracing a paleo diet?
Okay, so I'll give you the one sentence, right? When somebody says, "I'm doing the paleo diet, I want to do the paleo diet is great." You just ask them this question: which paleo diet? And if they don't get that, explain that, well, if we go back, deep in primate evolution, right, specialization on small fruits or insects may have been an important thing. If we go much later, there were hominins that were, well, do we want to eat the Paranthropus diet? Do you want to eat grass, maybe? So the diet has changed, right? And the reality is that that evolution, right, is happening always, right? So probably the best foods to eat in terms of being tuned to your biology are going to be foods from fairly recent ancestors that those ancestors would have eaten, right? So one of the things that paleo diet people say, oh, don't eat wheat, right? Well, if your ancestors are primarily from the Fertile Crescent, right, where there was early domestication of wheat, you probably have the genetic makeup to do very well digesting wheat, and wheat is probably like awesome, right? Paleo diet people also like to eat things like almonds, right? They're so totally domesticated, right? So, so yeah, there's some logic for eating a diet that is in tune with what your biology is, and what your biology is shaped by evolution, but it's shaped by very recent evolution too. Evolution's happening all the time. And you can figure out what you should eat and what is tuned to your biology, you know, based on a lot of different kinds of evidence. I don't eat soy, it gives me gas, right? I don't think it's attuned to, you know, either my gut microbiome or my genetics, right? I've figured that out. An element of the paleo diet is eating lots of protein and fat and meat, right? That can, in certain circumstances, turn out to be a reasonably healthy diet all on its own for different reasons, right? In terms of losing weight, but vegetables are also good. So yeah, so my answer is always which paleo diet.
A long time ago when I was a kid, we used to think of species as totally separate. Now we're saying the boundaries are a little fuzzy, and
Yeah.
species can interbreed in some cases. What are the major factors that separate species that they can't interbreed productively?
So I think the place to start is that, well, first of all, depending upon the group that you're talking about, what a species is becomes a very different thing. So if you're talking about bacteria, different set of rules. Plants, different set of rules. So if we go into mammals, right, we have a sort of specific set of rules. Speciation seems to happen when populations become isolated for some period of time that is significant, maybe long, maybe short period of time, and then they diverge. And they can diverge in a way that's fairly random, where their adaptations remain fairly similar and they look similar, but the divergence might lead to a genetic incompatibility in terms of offspring between those two groups, right? And so there's a significant kind of separation. So when they interbreed, at the very minimum, the offspring are less viable. You can also have, when species diverge, adaptation to make them look very different. So the obvious one is size, right? As size diverges, interbreeding is going to be less likely. Another obvious one is genitals, right? As they change and don't fit, you know, less interbreeding, right? You can have changes in chromosome number, and that leads to, you know, infertility in offspring. So donkeys and horses have different numbers of chromosomes, and so you get infertile mules when they do interbreed, and so there's a barrier, right? So the idea is isolation happens, evolution happens, and then that leads to some incompatibility in interbreeding. So that leads to then the generalized correlation that if things are different species, they don't interbreed, are unlikely to interbreed, don't produce viable offspring, as a general correlation. But hey, the world is like, there's no rule that says that has to happen. Things can diverge for a long time and still be capable of interbreeding. And then when they come back into contact and look different, they still interbreed. And there is a good reason to interbreed with the locals, right? To pick up their immune genes, right? So there are both reasons why species should not interbreed, right? Because the interbreeding might result in non-viable offspring, right? And there are reasons why it is advantageous, right? So that's why we start to see more and more these examples now that we can do ancient DNA, that integration and interbreeding and hybridization does happen. And hybridization can also be an opportunity to create a new species as a hybrid, you know, takes on its own fate.
There was a few years ago, a video documentary came out. It was called The Journey of Man.
Uh-huh.
I don't know if you.
I don't think I watched that one.
It was done by.
Who produced it? Was it a Nova one or?
Oh, it could have been, yeah.
Okay.
But it was done by biochemists who really studied the genes and basically would trace the journey of man from Africa. But one thing I think that he pointed out is that in the genetic makeup that they studied both in Africa and all different parts of the world, that they could actually see that some genes are much similar, like they would find some people's genes in Europe or some other places much more similar to some genes in Africa than, genes around that person's area. So is there any more clarification that has been brought since? I guess you were talking about markers or specific genes or something like that.
So there's a couple things that would be going on. So one, the general observation is there's more genetic variability in African populations than say there are in a European population. And that's just because the African population has had more time to accumulate variability, and that the migrant population that led to the European population was just a subset of African variability. And so then a variability that's accumulated, say, in the European population has had less time, so you're going to have less variability. And then if that population, right, migrates somewhere else, you again have sort of a new shrinking of variability and then less time for it to accumulate. So there's one model would be sort of a series of waves, right? And with each wave you get less genetic variability. Every wave retains genes from its ancestral population, going back to some that would be, in the African ancestral population. The other thing is, it's very, very hard to find samples of people where their ancestry is exclusively from one place. So, people migrate back and forth all the time. It's naive in the extreme to imagine that there's never a migration back the other way, right?
Can you tell us what
I want to give people first shots.
I just wanted a sentence or two on ancient DNA. I thought that was a real, that was hard. Is there good reliable data now?
Oh, ancient DNA. Yeah, so ancient DNA, it is hard, right? I am not an ancient DNA specialist, but there are these fabulous labs and fabulous workers who are continually improving coverage and ability to extract ancient DNA. So Ches, we hosted in the fall of sort of the Frontiers in Archaeological Science workshop, and one of the people that spoke was Vivian Sloan. She's just finished her PhD. She's like a total hotshot, and she is getting DNA out of dirt. So she has worked out a methodology to pick out bits of hominin DNA, has actually gotten hominin DNA. It's at a cave with a lot of hominin fossils, right? So we know that there would have been lots of DNA, but she's actually been able to suck it out of the dirt. And so the ability to extract it is getting better. Then computationally it has to be kind of assembled and put together. The ability to do that is getting better. Computing power makes a big difference. And so what we then get as we get an ancient genome is, what matters is coverage. How many times does a particular site get covered, right? So poor drafts are 1 or 1.5 times coverage. Good drafts are 30 times coverage, right? So that you have 30 different reads on a particular part. So our ancient DNA, they're all like drafts. They're like a book with some missing pages, swapped pages, but you can get the narrative out of it. So it's pretty amazing. I think that there's going to be lots of good stuff on genes and gene functions of ancient DNA that will be able to compare a gene for something with a modern one and get an idea of how it functioned differently. The population genetics of inferring migrations and stuff, that is hard and involves a lot of modeling and math and assumptions. And I'm not sure how much to trust all of that, but it keeps getting better. You know, so much so that I mean, it really is an amazing guide in terms of giving us a framework of what happened.
Okay, so we'll go for one more question and then we're going to wrap up.
Can you tell why, after the introduction of agricultural practices, the human dentition began to suffer? Because prior to that, it seems to be almost intact, and following agricultural diets,
So
it begins to show the same kind of problems that we suffer from today.
So we've had a long history of teeth sort of shrinking in size since sort of Homo erectus, right? So our teeth are smaller, okay, than ancestors would have been, so that's one thing. I wouldn't call that our dentition suffering. We do have various pathological indicators that appear on teeth. So like enamel hypoplasias or various metabolic insults that happen, interruptions in enamel deposition. That can happen because of a period of poor nutrition. We could argue in some agricultural circumstances, in some groups, that is more likely to happen. The other thing about agriculture is we can start to incorporate a lot more fermentable carbohydrates in our diet, which means forming caries producing plaques much easier, right? Bacteria feed on those carbohydrates, and if you're not brushing your teeth, doing dental care, you get cavities, and the dentition goes all to, cavities are not good. They'll kill you eventually. So I would say with agriculture, I would point the finger at fermentable carbohydrates.
Well, thank you. Thank you very much for speaking to us today.
Thank you guys very much.
And thank you all for coming. This nice crowd on a rainy day.